All right, so thank you very much for inviting me for this talk. So my name is Olaitan I. Currently, I'm the training officer for ASBCB. So that's African Society for Bioinformatics and Computational Biology. So the goal of ASBCB is to advance the knowledge of bioinformatics, primarily in Africa, but then we've been covering beyond Africa as well. Okay, so I will be presenting some of the visualization tools that we've developed through the Omics Codathons. And primarily, I'll be presenting Prostruc, but then there are some other tools that I would like to talk about. So the idea of Prostruc is to do 3D structure prediction using homology modeling techniques. As we know, UniProt contains over 250 million protein sequences, but then the validated structures experimentally determined in the protein data bank is just about 200,000. So there's still a lot of unvalidated protein structures, basically. So we decided to develop a tool. The goal of the 3D structure prediction tool, Prostruc, was not necessarily to be like the most accurate tool that would beat some of the known tools that we know that do well with 3D structure prediction. But we wanted to build a tool that the steps of the 3D structure prediction workflow can really be captured in that single tool. So homology modeling is basically a computational technique to predict 3D structure for a given protein based on similarity to one or more known proteins. And typically, the ability to accurately predict and model protein structures is very essential in different fields: drug discovery, protein engineering, functional annotation of genomes, and the likes. So we have existing homology modeling tools like Modeler, FireRate2, that do this work, but then some of them have certain limitations. Some of them require substantial computational resources, specialized software installations, or advanced expertise by the user, making them not that accessible to users, especially users from under-resourced areas. I represent people from limited resource areas or lack of funding areas for the most part. So that was what we're trying to do, to see how to have a tool that can probably just be easier to use, especially for people in that type of a situation. So we're to do a web interface and then also a command line interface for this tool. So some tools are designed to specialize in certain protein types. Those are some of the limitations we also realize. So there's need also for accuracy and precision of structural prediction, the need for open source tools, and just make it open access and not closed access, and then integrating the different steps of 3D structure prediction into one single workflow, which is where the issue of accuracy becomes challenging in that case, because ease of use becomes more of a goal of the tool than some other goals that we would have been pursuing. So just integrating everything as more like a one-stop shop for 3D structure prediction. So some of the things that we noted here on this slide: tools vary in their interfaces of use, learning curve, and things like that. So can we have a one-stop shop for all the steps of protein structure prediction? Okay, these are some of the objectives that we noted down using open source libraries, Python libraries like BioPython. Enhance structural insights, have a user-friendly web interface, have a command line interface as well, increased speed and accuracy. And here is a workflow for Prostruc. So the input to Prostruc is your target sequence from the user, which is typically a protein sequence in FASTA format. And then we do pairwise alignment using the pairwise module for BioPython. So, and we feed that into the Promo3 homology modeling engine after downloading the PDB file. and then do some type of validation with targeting low RMSD, which is typically what people do. And then we want a TM score that is as high as possible, closer to 1. And then we're also looking at human score as well. So we use all those to kind of just eliminate anything that is not as good of a prediction. And then we come up with a model. And then hopefully do visualization. So here is a slide that shows the sequence alignment results from Prostruc. And then this just gives a report on the protein analysis. So basically, we have the top 5 models returned, and we email that to the user. Now, you're going to see an example of how we capture your email and things like that shortly. Okay, so this just kind of explains again what we do in eliminating models and being able to just return the top 5. The main things we are using as metrics are the RMSD as low as possible, the TM scores as well as close to 1 as possible, and then the human score. So, and we also did some comparison with popular models like Swiss model, Modular, Promod 3. So for the command line interface for Prostruc, because Prostruc is available as a Docker image where you can download the Docker image and use it, and that gives you the command line access to it. Basically what you're doing is to call it with an example here like Prostruc. You provide you the argument dash sequence, and you could just copy the protein sequence there on the command line. And then you provide the argument for the job name and just put whatever name you want for the job, and then provide also the email to it. So it's installable through pip. Just do pip install Prostruc, and then you get that. For the web interface, you can upload the protein sequence and paste it into the protein sequence part, the amino acid sequence part of the web interface as well, and also provide your email address and submit the job. And you get an email telling you, dear Prostruc user, thank you for submitting your job. So that's when the job is submitted, you get an email notifying you that the job is submitted. And then when the job is completed as well, you get an email returning the models to you, as well as the just telling you that the job is done, and then you have the model. So we have a paper for Prostruc published in Frontiers in Chemistry, so it's there for people to read. And so here is just highlighting some of the contributions to the field, integrating 3D structure prediction into one single pipeline, doing protein sequence analysis, integrated homology modeling engine. And the main thing is just easy to use tool for structure prediction, which is customizable, scalable for large studies, and open source. So making it open for collaboration by the community. So future directions would be to increase the speed, integrate the visualization tool into Prostruc's prediction. Those would be some of the things that we could do. So the main highlight here are the free accessibility to researchers with varying levels of computational expertise, enabling high throughput structural biology research, and providing a flexible framework and user-friendly interface is something that just makes efficient structural prediction happen. So as part of my role as the training officer for ECCB, there are some other tools that we develop in these codathon events, or some people call them hackathons, and there'll be some tools that I would also like to highlight here. So here is the team that developed Prostruc. So Will Singh is the main developer. Shivani was the lead for that particular project, and you have some other co-authors like Nigel, Tohib, Musa, and Abdul Wasiu. And then I was basically just supervising the entire work itself. And by the way, these are mostly Africans, but Shivani is Indian. So the idea of the Omics Codeathon was just to increase representation of the life sciences in under-resourced areas where there is lack of funding and lack of access sometimes to knowledge and things like that. So there are some other visualization tools that we've developed are these Omics Codeathons. And these are virtual events, by the way. They're not like in person, bringing people to one particular site, because we don't have that money yet to do that. But hopefully at some point, we'll be able to get money to do more at the Codeathons. And if you have money for us, feel free to contact me. So we have this other tool, XVAR, and this is published in scientific reports. So what it does is enables you to visualize differential gene expression. You could also look at things like copy number variants, SNPs, and indels for your sample. So it has the R-Shiny component, but it also does the analysis. If you run XVAR on a Linux machine, you can also run some of the functions that enable you to do some analysis, which I will show here on the slide. So you could do your processing of the FASTQ, doing conversions, FASTQ to BAMs and stuff like that. You could generate counts for your gene expression data. The input is basically RNA data, right? So you can do things like differential gene expression analysis with it. You can call your SNPs, call your copy number variants, call indels on the analysis side. And on the visualization side also, it has functions. This is an R package to do expression visualization, SNP visualization, copy number variant visualization. The model organisms that are captured are human, mouse, yeast, C. elegans, and the other ones there. So here's the team that developed XVAR. So the team lead was Hiba. But then the main developer was Imran. And then Najla was also one of the bioinformaticians on the team, and I was just overseeing the entire work. We also have NeuroVar, which basically is an open source tool. It has a Python implementation, and it also has a dashboard that allows you to be able to visualize biomarkers of specific neurological disorders. And that is also published in gigabytes. You can read it. So here's the team that developed NeuroVar. So Hiba is the team lead as well, while Najla is the bioinformatician on the team, and then I was just supervising the work. Then we have this tool for genotyping rhinoviruses. It's a bioconductor package, so it's installable using BioC Manager. And what it does is the input to the tool is a FASTA sequence, and then it tells you the strain of rhinovirus that you have. So here's the team. Actually, I think the team was swapped. Sorry. Okay. So that's not the team that developed Rhinotyper, actually. That was a mistake there on that slide. So I'm going to move on to another dashboard, which is Rare Insight. And this is also published in scientific reports, so it's there available to read. I'm going to tell you more about the Rare Insight dashboard shortly. So basically what we wanted to do here is to have a dashboard that allows you to generate customizable clinical reports that can enable the clinician to make informed decisions about rare diseases from VCFs, and also enables the patient who might not really be savvy with, looking at VCFs for sure, and even caregivers. So our goal was to deploy a dashboard that would allow for multiple input types, VCFs and the likes, and then you can filter and search your VCF and look at certain things using the dashboard from your VCF. So it uses the NF Core rare disease pipeline to do that, the analysis part of it in generating the VCFs, and then we have a Shiny dashboard component to actually look at things from the VCF. The contributions of this work to the field are providing genomic literacy through tutorials. It's open source, so it's collaborative. Compared to existing dashboard that we kind of benchmark with, offers a level of independence for patients in understanding their diagnosis. Also targets caregivers that might not really be very savvy with looking at VCFs or even patients at all for rare diseases specifically. So here's the workflow for that. So you have the NF core rare disease pipeline producing the VCF, which you then feed into your R-Shiny, and then you're able to look at things in the dashboard. So this is not suitable for novel unreported variants for rare diseases with no known annotation in ClinVar, because it uses ClinVar basically as its source of truth. And not yet able to export results to a clinical report. We're still working on that. So here is what the dashboard looks like. Kind of just telling you about rare insights here. But then it has some kind of tutorial glossary telling you a little bit about DNA and things like that. What is a cell and things, just in case the users want to learn some things about genetics. So it allows you, basically it can take whole genomes. It can also take, the pipeline can take whole genomes or whole exome as two types of input data. Then it just collects some information from the user. So that's what this page is about. And then you have the search panel. You could actually type a gene name and then it searches through the VCF to kind of tell you things about it there. So here is a page that just shows the VCF panel. And this is just, general information for patients about disorders and things like that. Information for clinicians and researchers. So here's the team that developed Rare Insight. So Kimberly was the team lead for that project. And then we had other people like Firas, Eva, Gideon, who were bioinformaticians supporting that. And these are all Africans, just to highlight the fact that the representation of under-resourced places like Africans in the life sciences is being achieved through this. And we have a paper, for instance, in Scientific Reports for this. So for my acknowledgement, all this work would not have been possible without people that support the work, like George, Shivani, George Chenemi, Kimberly, and support from the NIH DSS. Sometimes we have facilitators from the EBI actually providing workshops to us at these events, the hackathons. Biodata Sage sometimes provide support in terms of resources. I have colleagues like Derek Raymond, Amna Siddiqa, Eric Ways, Justin Chow that help in listening to presentations by participants of the hackathons when they are presenting their work. And here is my email if you have inquiries about the Omics Hackathons or for a job opportunity if you have a job for me. Thank you very much for listening.
Thank you, Olaitan. So do we have questions?
Are there specific countries that are playing more of a role within this organization, or is it all across Africa? How is the distribution, demographic distribution?
So thank you for that question. So in terms of demographic distribution, it's all across Africa. But from what we've seen from participation, we have certain African countries at the top of the bioinformatics list, if you will. So I would say countries like South Africa are usually at the top of the list in terms of activity and performance. Then we have Kenyans also attend a lot and actually contribute actively. Then Nigerians, Ghanaians also usually at the top. Some of the other countries, Morocco and Tunisia also usually are quite active. But then we have some other countries in Africa coming up. But in terms of other non-African, we also have participation beyond Africa. People from the UK, Canada, Mexico, the Philippines, and the likes actually attend to. Yes.
Yeah, my question I think was quite similar to that, although I didn't hear exactly what the question was. So were you describing the users of your tools? That, like, where are they from?
So the ones I just described are the participants who are attending the hackathons and developing the tools.
Oh, right.
So they attend these hackathons. We organize workshops in the hackathon. So we're teaching like running, using tidyverse, developing code with R, Python, Nextflow, SnakeMake, and the likes, learning to use GitHub. Then they query public data sets, pull down data from,
Right.
yes.
So my question is about, these are nice tools that are accessible to different types of users, not
They
just by
are all open access. They're
And-
accessible to everybody.
And who do you find are using them? And I was also wondering, how can you make things more accessible? Like, all these things seem to be in English. Is that a problem for your user base?
Thank you very much for that question. So it's something we hope to get to, but one of the challenges we've had is that sometimes we need to actually communicate with people that actually speak French, even in terms of participation, even apart from user base. The user base currently are mostly, like Africans and people that attend these events, but it's open to everybody in the world. But sometimes in participation, some of the people attending are actually speaking French. So it's one of the things we need to do now is just have like interpretation and things to also serve more people that are even French speaking or Spanish speaking attending these events.
Okay, so please join me in thank you, Olaitan, again. And
Thank you very much.